Hello, I'm Sherry Flint-Garcia. I work for the USDA Ag Research Service in Columbia, Missouri, located on the University of Missouri campus. Today I'll be telling you about corn genetics, the history of maize. Where does our food come from? Not from the grocery store. Our food today is the result of thousands of years of hard work. Starting around 10,000 years ago, human populations began to transition from hunter-gatherers to early farmers. And they were trying to develop stable and productive food sources since those populations were less mobile than they were. And so they began to tame or domesticate the plants and animals around them. In plants, people selected for plants that were easier to harvest or had bigger fruits or seeds. Here you can see a number of examples. We have rice. Wild rice species tend to shatter their seeds and spread the seeds. The wheat head is much more compact, and the Q allele allows for easy threshing of the grain, whereas wild wheat holds on to the chaff around the seed. In tomato, you can see that there's a large increase in the fruit size. And in the case of sunflower, wild sunflowers have many, many more flowers, which flower over a longer period of time, which makes it harder to harvest the seeds as they mature over different times. Clearly, seed size was one of the most important traits that was targeted during domestication. The same is true for maize or corn. Corn was domesticated from Teosinte. The wild species is called Zea maize, a subspecies called Parviglumis. And it's adapted to the lowland growing conditions of Mexico in the states of Jalisco and Guerrero. And beginning about 9,000 years ago, people began to domesticate Teosinte to form early primitive corn. There are major morphological differences between Teosinte and corn. You can see that the plant is very highly tillered or branched or bushy compared to modern maize. And you can see that the plant does not produce true ears like we know in corn today, but rather the seeds are stacked on top of each other so that when the seeds are dry, the connections dry, and the seed can shatter and spread like a good weed. In addition, the Teosinte seed is enclosed in a very hard fruit case that surrounds the seed. And so these hundreds of little tiny ear structures with their very difficult seeds that are enclosed in these fruit cases on this highly tillered plant was very hard to harvest and to use as a crop. And slowly over time, people began to domesticate it by selecting the traits that were important to them to slowly transform it into the corn that we know today. The different those different traits that are in Teosinte and maize are controlled by different genes. And scientists are studying those genes and have identified a number of the genes to date. One of the most important is called Teosinte glume architecture, and it controls that hard fruit case around the seed that is not present in our modern maize. The Teosinte seeds are enclosed in this hard fruit case, and the maize kernels are considered to be naked. And this gene, Teosinte glume architecture, mutations in this gene opened up the fruit case to make the seed accessible and allowed the seed to grow bigger over time. A second trait that was really important was tillering, or the branching structure that's different between Teosinte and maize. The gene responsible for this is called Teosinte branched. You are all familiar with corn. It usually has one main corn stalk, and the side branches are short and they end in an ear. In Teosinte, there are many, many tillers or branches, and they often end in a tassel, the male part of the plant, and have many, many, many ears along the branch. And the final gene that people have studied more recently is called grassy tillers. Maize generally has a side branch that ends in a single ear, whereas Teosinte has side branches that has multiple ears. And so you can imagine if you were a hungry early farmer, you would try to target each of these different traits to make your Teosinte, now possibly a more primitive maize, easier to harvest. Another trait that's really important to understand is photoperiod sensitivity, or day-length sensitivity. You can see here I have colored the different air regions of North and South America based on their day length during the summer solstice. In the Northern Hemisphere, the farther north you move, the longer the days are. So that in Canada, you have upwards of 16 hours or more of day length. Whereas as we get closer to the equator, the days are much more equal, 12 hours of day and 12 hours of night. Because Teosinte is adapted to southern Mexico, it's adapted to regions that have closer to about 12 and 12, or more like 13 hours of day. And so when we move Teosinte from this region of adaptation in southern Mexico up to Missouri, for example, the Teosinte is not adapted. It's not getting those signals to initiate flowering. And here you can see an example of what Parviglumis, the Teosinte, looks like. This picture was taken at the end of September, and Teosinte is not flowering at all. And we have a sample of Teosinte that has been modified to not respond to photoperiod or day length. And in this case, this Teosinte is able to flower in August in Missouri. And so you can see that this day length or photoperiod sensitivity is a very important trait that we'll be coming back to many times today. I've oftentimes wondered what it would be like to go back in time and to see all these traits changing over time. But always the risk of going back in time is that you may change something and change the future. But at the end of the domestication process, we still did not have modern corn. We had primitive maize, a little tiny ear that maybe had 30 or 50 naked kernels. These seeds were small, like a popcorn kind of seed, did not have a seed coat. The plants had fewer ears and many fewer tillers. This probably took about 300 to 1,000 years to get from Teosinte to primitive corn. So it's a very slow process. Once primitive corn was developed in southern Mexico and people left that center of origin, they took their primitive corn with them as they moved north and south across the Americas. Every time the corn ended up in a different environment, it was able to adapt. These adaptations result in land races or locally adapted populations. And there's a lot of genetic variability from plant to plant within each land race. And this variability allows the corn back then and still continuing today to adapt to different environments. There are many different kinds of environments that Teosinte or primitive maize would have encountered. For example, high dry plateaus, maybe adapting different tolerance mechanisms. If it was adapted to humid coastal regions, perhaps it adapted with disease and insect resistance. Perhaps different soils and temperatures, different day lengths. That is a topic we've already talked about. And different elevations. I'll be talking about elevations quite a bit very soon. But another different environment is human use. And this includes various food uses and ceremonial purposes. So by the time Columbus sailed the ocean blue in 1492, corn was already growing from Canada all the way down to the southern tip of Chile. And Columbus and other explorers brought the corn back to where they came from and other places around the world, where it continues to adapt today. On our next slide, we will be starting a tour of land races from across the Americas. These are all naturally adapted, open pollinated populations, and they each have different interesting characteristics. So as I mentioned, Teosinte was adapted to a very specific day length, and the same thing is true for land races, these adapted populations. Here we have a sample of a number of land races that are found from across the Americas, ranging from North Dakota on the far left, all the way down to the southern part of Chile on the right. And you can see that for each land race, I'm showing you where it originated, the name of the land race, and the latitudinal degree. So we will begin our tour in Dakota. Assiniboine is a very interesting land race. It is not happy in Missouri. It's adapted to the shorter season with longer days that's present in North Dakota. So in Missouri, it germinated, it grew, it flowered very quickly, and it was mature and dead well before the middle of August. If we had grown it in North Dakota, perhaps it would be taller, later, and happier than in Missouri. We're going to eventually move south across the continents, moving across the equator, and we'll continue south. Notice that the plants are getting taller and taller as we move closer to the equator. These land races are adapted to the shorter days, the 12 hours of day and night that is present at the equator. And so they are waiting for the right environmental signals to initiate flowering. And in the meantime, they're just going to continue to grow and grow and grow until the short day length signal is received and they begin flowering. Once we cross the equator, then the corn plants become shorter and shorter and shorter. The different land races are now adapted to ever increasing day lengths, just like the long day lengths in North Dakota. As we move south across the continent of South America, the day lengths get longer and longer again. All these different land races that are adapted to the equatorial regions here would be much shorter and earlier flowering if they were growing in their original adapted regions. And each land race has different distinct characteristics: plant morphologies, ear shapes, kernel colors, grain textures. Some of them are very fun with spotty and stripy kernels. And after we cross the equator again, the plants will begin getting shorter and shorter as their native day lengths become longer and longer and closer to our environments here in Missouri. By the time we get down to the temperate zones of Uruguay and the southern parts of Chile, those growing conditions are very similar to the Corn Belt in the United States. So it's clear that we can't just any land race from someplace in the Americas and grow it in Missouri because they're unadapted. But the question is, how far can we push the limits here? How much work will it take to try to rescue some of the desirable traits that we know are present in the land races? We can obviously use land races that are from the farther north and the farther south regions, but how far into this crazy wild jungle here in the middle can we go to find some interesting genes and alleles? This is the exact same picture. The first picture was taken in the middle of September. This picture was taken on October 8th. And you can see we're farther along in the season and most of the corn is drying down. But those land races that are adapted to the more equatorial regions are much later. And you can see that there are very many signs of unadaptedness. Many of them are too late. Here we can see pictures of some interesting fungal diseases like a smut fungus. If you like mushrooms, this is actually quite tasty. Sometimes on the land races, the ears are very high up on the plant, and sometimes the roots are very bad, so that the corn plants will either fall over because of the weight of the ear or because the roots are bad. And when the ear lands on the ground, it will rot and die. And sometimes there are some land races that are just plain bad. They're way too tall, way too late. They don't make an ear because the male and the female are not synchronized. And you have all these different disease problems in addition. So in addition to being adapted to different day lengths, land races are also adapted to specific elevations. When we drive around in Mexico and South America, as you go up into the mountains, suddenly you'll notice that all the corn stalks are purple in the ear zone. And here are some land races that are adapted to the highlands, showing here in the picture, and you can see the purple corn stalks. And you can see the stalk pigments that are in the ear and the stalk in that area. And there are also macro hairs or hairs that are on the corn stalk as well. These are clearly adaptation traits, but the exact mechanism for how these traits confer adaptation is really not clear at this point. And so we have a grant to study highland adaptation. One of the first adaptations I told you about earlier was to environments that were in highland. So primitive corn moved from the lowland site of domestication, where Parviglumis was growing, and it started to climb the mountains into the highland regions of Mexico in that area. This involved gene flow or crossing with other different Teosinte species, one called Mexicana, that is adapted to the highlands. And this Mexicana Teosinte also has purple stalks and hairy stalks. And once corn moved down into South America, corn also gradually climbed into the Andes. And so even though the Teosinte, the highland Teosinte that I just talked about, is not present in South America, and it didn't contribute to the highland adaptation in South America, the corn was still able to adapt to the South American highlands. But it also has those same pigments and hairs. Here you can see the purple stalks in the Mexican land races and also in the South American land races. So how did this corn gain the same adaptation traits in the absence of the Mexicana Teosinte? And so we believe that this is an example of parallel evolution. Interestingly, some of the best tasting traditional food corns from South America and from Mexico come from the highlands. In the Mexico City region, a very famous landrace for eating like sweet corn, corn on the cob, but it's not a sweet corn, is called cacahuacintle. It has a very interesting kernel texture that has a very good flavor. And up in the highlands of South America, there's a landrace called Cusco. It has very large seeds that is used to make corn nuts. These landraces are not really adapted to Missouri. And the question is, can we try to recover these flavors and textures somehow in our US corn? So when we talk about breeding and how we come to modern corn, we really don't always understand plant breeding. And so I'm going to use a dog analogy. I think a lot of people understand dogs. So when you talk about a mix, for example, a beagle mix, you know that there are certain characteristics in the mix, but they don't all look exactly the same. And then there are kennel clubs out there that breed specific breeds, for example, the pug and the beagle. And when people talk about pugs and beagles, they know that there are very distinct characteristics. And nowadays, you can cross pugs and beagles together to get crossbreeds like the puggle. Corn has the exact same type of individuals. You can take land races or breeding pools like Reed's Yellow Dent and Lancaster Surecrop, and you can derive pure breeds. So these land races are basically mixes, and the pure breeds are called inbreds in maize. And you can see very distinct characteristics that define what B73 is. Many, many small kernels in a very upright leafed plant compared to Missouri 17, for example, has larger seeds, and the leaves are much more horizontal. And then, just like in the dogs, we can make crossbreeds that are called hybrids, like the B73 by Missouri 17 hybrid. The hybrid is taller and makes a bigger ear than the pure breeds. And that is the reason why farmers do not grow heirlooms or land races in the United States anymore. Beginning in the 1950s, hybrids became available, and they were widely adopted in the 1970s and nearly completely displaced all the land races in the United States. And so we have to try to understand how the breeding process works. So we've used the dog analogy to understand the terms of land races, inbreds, and hybrids. So how do we make an inbred? For example, if we take Lancaster Surecrop, this is a land race that has a lot of variability from plant to plant and ear to ear. You would usually choose the better of the ears to proceed with your breeding program, and you wouldn't waste your time making something that is substandard. But if you take the pollen from the tassel and you put it on the silk of the same plant, that's called self-pollination. And if you self-pollinate a number of times, for example, 8 times, you become very much of a pure breed. And taking Lancaster Surecrop and selfing it 8 times led to the development of C103 from Connecticut. And this is what they consider as an early cycle inbred. And you can do this for any of the different land races. And this is how early corn breeding began. The corn breeders in the public sector and the private sectors have really worked over the last century to develop a system of making hybrids in the United States. And there are certain heterotic patterns that make hybrids more predictable. And you can see here that inbreds can be roughly organized into 3 different groups: the stiff stalks, the non-stiff stalks, and the iodines. And the strange names results from early efforts to try to improve the stalks of some of these land races, thereby making them stiffer. And so if you know what group some of these inbreds belong to, you can predict what kind of hybrids you make. You can't really take 2 stiff stalks and cross them together to make a productive hybrid. But if you take a stiff stalk and you cross it with a non-stiff stalk, chances are much better that you'll be able to make a good hybrid. And the seed industry has taken the public lines that were developed in the 1950s and the 1970s, and they continue to make improvements. A lot of those early inbred lines were developed by directly selfing from a land race like we talked about earlier. But now industry is very much focused on trying to maintain these patterns of the stiff stalk, the non-stiff stalks, and the iodents. For example, if you're breeding for northern corn, the iodent germplasm pool or inbred line group is much more tolerant of cooler temperatures. And so you would try to continue to maintain iodents and perhaps cross them with non-stiff stalks or stiff stalks to make hybrids. And so modern corn breeding really focuses on trying to maintain this pattern. And so you may take two of your better performing stiff stalks to cross those together to start making a population that you can derive a new inbred line or purebred from. And at this point in time, there are likely more than 1 million different inbred lines that have developed in the private and public sectors over the past century. And so understanding how they're all related together can help you predict how good of a hybrid they are going to make. Not all hybrids are equally productive. You can't just cross any two together, like I mentioned earlier, and expect the hybrid to be more productive than the parents. And so here you can see a number of examples. The first panel shows B73, which is a very common inbred line. It was very important in the 1970s. And if you cross it to something that's very closely related to itself, LH195, even though they don't really look the same, they're very closely related, they are very closely related, and so their hybrid doesn't produce a whole lot more than either one of the parents. And so that inbred hybrid combination is said to have low heterosis or low hybrid vigor. If you start to make the parents more distantly related, the better your chances of making a good hybrid are. So when you cross B73 with LH82, which is much less closely related to B73, you can see the hybrid makes a very much bigger ear than either one of the parents. And so it's said to have heterosis or hybrid vigor. And this hybrid outperforms either one of the parents. And again, as the parents become less related to each other, you can expect more heterosis. And so you can see here Missouri 17, which is very unrelated and very different from B73. You can see that in the ear morphologies. The hybrid is even better than we had before. And the B73 by Missouri 17 hybrid was one of the best-selling hybrids in the 1970s. And as I mentioned before, industry has taken some of these public inbred lines, like the one that was developed by the University of Missouri with the USDA, and they continue to improve these different varieties. So they've made an improved version of Missouri 17 called PHZ51. And when you cross PHZ51 by the same B73, the hybrid is also very good. It doesn't look like it's that much better than the Missouri 17 hybrid, but it's not just what the size of the ear looks like. It also has to do with how the population performs in a farmer's field. It's likely that PHZ51 by B73 is able to tolerate higher planting densities, and thus you'll end up with a higher yield on a per acre basis, not on a per plant basis. And so the ultimate goal of plant breeding is really to improve each of the parents, B73, Missouri 17, and then test all those different hybrids to try to find the best hybrids. And biotechnology is just another extension of plant breeding. When you're doing plant breeding, you're restricted by crossing corn with corn, or perhaps corn with teosinte, if you're willing to go that far out. But biotechnology allows new opportunities to introduce genes from other species into corn, for example. And these traits can be created by using transgenic approaches. It is really important to understand that even though an ear of corn looks okay, for every 3 kernels that you lose per ear, that results in a bushel of corn lost per acre, or $3, based on a $1 or 1 bushel costing $3, a $3 loss per acre. So imagine if you have 1,000 acres of corn, if every ear is just missing a few kernels, that's $3,000 lost. And so anything we can do to prevent the loss of those kernels, for example, using GMOs, we can make farming more profitable. And it's important to note that transgenic crops can offer several different advantages. They can allow the corn plants to become disease and insect resistant, drought tolerant. You can be more nutritious and potentially even yield more at harvest. And here you can see by introducing a gene from a bacteria, the gene is called BT, It's a crystalline protein from Bacillus thuringiensis. The BT, a crystalline protein introduced into corn, makes the corn insect resistant, whereas the plants that are not GMO, that do not contain this crystalline protein from the bacteria, they've certainly lost 3, 6, 9, 15 kernels per year. And so you can see how this introduction of this single gene can have a very large effect on the profitability or the reducing loss for the farmer. It's also important to know that these genes are just DNAs that make proteins that are not going to harm human beings. And so the human body can't tell the difference between the BT and the non-BT corn when you eat it. And so research and breeding continues in both the private and the public sectors, the seed companies, the USDA, the universities. We are all very interested in understanding how all this selection, going from Teosinte to Landraces to modern corn, how that breeding process works, what genes have been responsible for all these different changes, and what trade-offs were made when we were selecting for higher yield. You know, some very important questions out there is, can we continue to make improvements in productivity and nutrition and feed the increasing world population in a sustainable fashion? I think that's one of the really important things for us to think about as we move forward. I don't work on corn alone. I have a great team of people that work with me here in Columbia, as well as collaborators that I'm on with different grants and different projects. And so I have 2 different technicians, Jim and Susan, my postdoc Nancy, graduate student Abby, and 2 different undergrads that work with me this summer, Melanie and Sarah. On our Highland Adaptation grant, Jeff Rossabara is the lead man. He's the boss. Jefe is the boss in Spanish. And a number of great collaborators from Iowa, North Carolina, Penn State, and Davis. And then more recent work working on food corns, which I didn't have the time to talk about today, is a collaboration with Jim Holland, who's with USDA in Raleigh, North Carolina, and his student Matthew Smith. I've been very lucky to have funding from both my home agency, USDA Ag Research Service, and NSF Plant Genome. If you have any questions, please feel free to contact me.